All right. Hi, everybody. I guess it's 10 o'clock, so we can get this started. Welcome to today's session of the Escarpment Labs webinar series. This is day 3 of EscarpmentCon. So today we'll be talking about quality control. Escarpment Labs is a yeast producer based in Guelph, Ontario, and we work with all breweries across the world to solve fermentation problems and create new flavor profiles. I'm Alex. I am currently in a business development role and project management role at the company. But I'll be the moderator for today's webinar. Today, our topic is cell counting, and I'd like to introduce today's presenter, Chris Saunders. Chris is a quality assurance technician where he focuses on ensuring that product reaches professional brewers across Canada. He has a background in software, which he has used to help improve and automate business processes, and his favorite beer styles are German wheat beers and Bavarian and Bohemian style lagers. For those joining us live, thank you, and feel free to start a discussion in the Crowdcast platform, as well as ask questions and upvote other people's questions that you're most interested in. I'll do my best to monitor the discussion as it goes along and follow up with our speaker if needed. For those tuning in live after the cast, check it out on YouTube when it gets uploaded, probably tomorrow. We've been pretty good in the last few days about uploading them. And thank you for checking out what we have to offer during this time. Without further ado, let's give Chris the floor and he can tell us about cells and how to count them.
All right, so first things first, just to make things a little bit simpler, I'm going to boot Alex so that he's not taking up valuable screen space. And we'll bring him back at the end. All right, so let's get started. This one, yes. Okay. So we're gonna talk about cell counts, and we're gonna go over. Oh, hold on, I need to make it full screen I need to focus that. All right, close that, maximize that. All right, sorry. Okay, so this is gonna be a live cell counting demo. I'm also gonna be covering, I guess, theory and the practical aspects of doing cell counting. And as mentioned, I'm Chris, and Alex is our moderator. So who we are, that was kind of discussed already. We're a Canadian yeast supplier offering fresh, locally made, and ready to pitch yeast. We provide a wide range of services across the brewing industry, including micro quality control, custom yeast bacteria development, and strain banking. And we also strive to be excellent sources of knowledge, and our focus as a company is on quality and education, as we're showing with this conference series we've been running. So previously I was a lab technician for Escarpment Labs. I still am doing that now, just to a lesser role. And now I'm in a business development role, and I've helped Escarpment troubleshoot cell counting issues, including clumpy group yeast strains such as Foggy London Ale. And this is a sort of a re-presentation of a talk that Alex and I gave to the MBAA District Ontario back in 2019. So let's talk about the background. So pitching rates, yeast is responsible for converting simple sugars into alcohol and carbon dioxide. For optimal beer production, it is important to provide yeast with the best possible environment for growth. Typical pitch rates from us are 10 million cells per milliliter, assuming a 10 to 12 degree Plato wort. And knowing the correct pitching rate and viability of your yeast is essential to making consistent products with predictable fermentation times. So this diagram here is just showing sugar will take in, yeast will take in sugar, ferment them, convert that into ATP and energy, and then they excrete alcohol and CO2. So yeast pitching rates. Determining your pitching rate is extremely important. However, there are some considerations to take into account before pitching. Lower pitching rates will often result in more flavor production, so esters, higher alcohols, and so on. Higher pitching rates are needed for more stressful environments or where ester production may not be required. So some examples of that are lagers and high gravity beers, etc. With lagers, we generally don't want those esters produced, so we'll throw more yeast in there. And also they're fermenting cooler, so we want to provide more yeast as well. Pitching rates are an approximation, especially when you're doing counts yourself. Depending on your technique, you can generally see a variance of about 5%. So just a background about pitching rates. So these are some of the esters and flavor compounds that can be produced whenever yeast are fermenting and whenever they ferment warm and stuff like that. So we can get ethyl acetate, which can be pear drop, and at the higher concentrations, nail polish remover. Phenyl ethyl acetate, which gives us raspberry and red fruit. Ethyl caproate, which can give us pineapple. That's the pineapple thing is pretty common with a lot of non-standard yeasts such as Brettanomyces. Isoamyl acetate, that synthetic banana thing. Kind of think of amoxicillin is a good one if you want a good example of that, or those cheap banana marshmallow things. Isobutyl acetate gives you sweet and fruity characteristics, and then ethyl caprolate gives you fruity and floral flavor characteristics. So a background on pitching rates. Some commonly asked questions about pitching rates and performing cell counts are, when should I perform cell counts and why should I perform cell counts? So anytime you're going to be pitching yeast, for example, from a keg or from a fermenter, or you want to monitor the yeast growth after pitching, so 24 to 48 hours. And why should I perform cell counts? That's to monitor yeast health through the repitching process and to supply proper yeast density into wort. Why should I perform cell counts? It's critical for many trendy beer styles. So lagers, too little yeast, high risk of off flavors. Hazy IPAs, too much yeast or unhealthy yeast can result in protease activity, which can result in the breakdown of haze. Some people, like, if you want to get those Iceman pours, the lack of foam can help you get there. But personally, I really like foam, and I think foam is really nice. There are other things with those hazy beers that can be responsible for reducing foam production, but mistreating your yeast is another good reason, which can cause decreased foam stability and foam in your beers in general. So what should you perform your cell counts on? Harvested yeast to determine your pitch rate for your next brew is probably the most obvious one. And on new batches of yeast to confirm. This is to confirm information provided from your supplier. So and another thing could be if you're rehydrating dry yeast or if the information isn't made apparent. And then how should I perform cell counts? Well, that's what we're here to show you. So your equipment and getting started. Before you can perform a cell count, you need the following equipment: a microscope with a total magnification of 400x and a movable stage. You can get microscopes, like you can get lower cost microscopes that don't have movable stages, but they are difficult to work with. You can eventually figure out how to move them around, but it's not a very enjoyable process, and it can be pretty tough. So getting a quality scope with that movable stage, and you need at least a 400x magnification, a quality hemocytometer, which we'll be talking about soon, a clicker or counter. You can use a counting app. Those do work. They're not great, and sometimes you can miss hitting them. So if you do want to use a clicker app, there's a bunch on the various phone app stores. But you can get a clicker, like a hand clicker that you just use for like counting people entering a business or whatever, for about $20. Erythrocin B stain is the one that we use internally. Or you can use methylene blue or trypan blue. We've removed trypan blue from our counting procedures. Trypan blue is a mutagen, and we also needed to implement a bunch of special disposal procedures for that. So we removed it to simplify our process, simplify our handling, and also trypan blue. If you are going to use that, absolutely make sure that you're wearing PPE, because that can hurt you. You'll need a pipette pump and disposable pipettes. You can use, so I'll be using this like cheapo pump I got off Amazon for $10. These work. I was setting up a sample, and they are tough to use, especially if you're loading. The hemocytometers need to get like 10 microliters of sample. It's really hard to load 10 microliters onto this, so you can end up over flooding your counting chamber. So what we suggest is micropipettes, which are quite a bit more expensive, but they are more precise and they're easier to use. Kim wipes, you can use for your counting chamber, you'll want to use Kim wipes, but you can just use for other things. You can just use paper towels, and then test tubes, plastic or glass. We use plastic ones because all the work involved in cleaning test tubes is a huge pain. So we just get reusable Falcon tubes. The ones we get are from Frogabio. They're affordable lab equipment. They're affordable in the sense that per unit they're fairly cheap. I think they're like 5 to 10 cents each, but that's per sample, and you have to buy quite a large amount of them at once. So depending on how often you're doing your counts, using disposable equipment like that is probably the best thing. But if you're doing counts less frequently, like let's say you're at a homebrew level, maybe going with glass is something you'd want to consider. Just keep in mind that glass can break, so it's something to be aware of. So equipment and getting started. Purchasing equipment can be an expensive upfront cost, but it's worth the money to prevent lost batches due to off-flavored beer. So here's just a breakdown we put together a year and a half ago. So the cost to set up, and this is a pretty bougie cell counting setup. So a really good quality microscope will set you back $1,500 to $2,000. A lab quality pipette pump is $400 to $800 each, with or you can get a disposable 1 mil one, or you can get disposable 1 mil pipette tips, $25 for $500. So you'll get a bag of them, and they'll generally look like this. You can work with those, and then you'll also want to be getting larger ones as well, 10 millimeter pipette tips, so you can actually pull out your sample and then work with it. Plastic test tubes, $80 for 500. Erythrocin B, $35 for the raw materials from Sigma. We are trying to work on putting together something for people to just buy pre-made erythrocin B. And then Kim wipes, which is $135 for 30 boxes. You can buy these on Amazon, but buying them on Amazon will come with a markup. So you could buy 1 or 2 boxes, but they will be drastically higher than the cost of 135 divided by 30. So the total on the high end for a pretty nice setup is $3,300. Now we can compare that to a lost 50 hectoliter brew of a really simple pale ale with just pale, 2-row, and Citra. So the malt, 60 bags or 1.5 or 1,500 kilos at $25 a bag is $1,500. The labor, 2 brewers, 2 days, 8 hours each, 32 combined hours at $18 an hour is $576. Yeast, which is, this is dated, so the price for our yeast has actually gone up. $1,170 for 50 hectoliter pitch of Escarpment Labs yeast, and then $100 in hops, which is about 50 kilos, and that's a rough estimate. And in this, we're excluding things like water consumption, rent, heating, energy, and all that kind of stuff. So on the low end, we're looking at $3,300 for a really, really simple blonde, like hopped blonde or pale ale. So if this goes bad, and then there's all the value on top of that that's lost. So just in raw material cost, we're at roughly the same, and that's just one batch of beer. Where with this equipment, you can get several hundred, uses out of this single cost that you purchased for, your cell counting setup. So your equipment and getting started. What is a hemocytometer? It's a counting apparatus that provides an easy matrix to count various cell types, including yeast. To view the yeast on the hemocytometer, you need a microscope of at least 400x with a total magnification and movable stage, as we mentioned earlier. So this is in this diagram here. So the upper one is just what the counting device looks like, and then in figure A, what you see there is this is the counting region, and then it's broken into 5 big areas, and then there's that small center area. And in that small center area, we want to be counting 5 quadrants, which is the upper left, upper right, bottom left, bottom right, and then the center. And then we will use really simple averages to kind of figure out what. I'm just going to close the chat here because it's really noisy. Using those, we'll count those areas, and then we zoom in onto each of those and then count them. Once you have all the equipment, you can start performing cell counts. harvested yeast samples are typically too dense to count and load onto the hemocytometer, so you need to dilute them. And we're going to go over that in the following slides on how to load and count a sample on a hemocytometer. Another thing to keep in mind here is whenever you're buying hemocytometers on Amazon and eBay and stuff like that, it can be pretty tempting. You can find these really affordable hemocytometers on Amazon. They'll usually cost you maybe $15 to $25. On the slide here, we have a comparison of what those look like. So on the left is a high quality hemocytometer, which I think is the one from our lab, or one of the ones in our lab, which cost us $125. I think they might be even more expensive. And then on the right is a low quality one that is, you can use it, but one of the problems is the grids are a lot lower quality. The contrast is a lot lower. And I've had to do them. One time we had our high quality hemocytometer broke, so I had to use the low quality one. And you will, if you are doing lots of cell counting, you will get really, really bad eye strain from this. And also, depending on what kind of scope you have, you can also end up with, you can't even see the lines, or you have to like tweak it so you can see the lines, and you have to tweak it back so you can see the yeast. So you can never see both at the same time, which makes the counting even harder. So definitely, if you're going to be buying a piece of equipment that you do spend money on, spend it on the high quality counter. Your eyes will thank you. I've done like an hour to an hour and a half of cell counting after a harvest, and it's resulted in I'm like extremely exhausted. My eyes are extremely strained, and you don't want to do that if I just use a regular. And it also slows you down. If you use a high quality counter, you spend less time fidgeting, and you'll be able to just get through, blow through your counts super fast. So a counting chamber demo. I've already loaded stuff onto the chamber. I can and I'll show you how to navigate around. So I have, there's this other person in here who is Saccharomyces snap, which is me sharing cameras from my computer as well. So what I'm going to do is I'm just going to swap over to one of those cameras. Actually, I think oh, hold on here. Technology's really tough. All right, so I'm just going to turn my camera on. I'll turn on this, and then we'll just let's see what's happening. Come on, do your thing. Okay, there we go. So that is loading. Save. Okay. All right, there we go. So we are connected, and we can see. So what I've done here is I've loaded on. I've loaded two samples. One is a sample of extremely concentrated Cali ale, and then I'll move over to the other sample, which is pretty dilute English. So this is a 1 in 1. Both of them are 1 in 100, but this is Cali ale that I had pulled off that I was packaging yesterday, and I pulled it off and threw it into a tube. And as we can see, this is what. Oh, yeah, you can still see me. This is what extremely concentrated Cali ale looks like. Even at a 1 in 100, it's impossible to count. And I'm just going to get this in focus. Oh. Okay, hold on. I'm like dealing with two cameras here. So, all right. So this looks good. Okay, I hope you can see this. Anyway, what we have here is we have our counting chambers. I'm just gonna pan around a little bit. So I would not count this. I wouldn't even try. I would put this underneath the scope, be like, this is way too concentrated. I'm going to further dilute it down so that I can actually get to a workable solution. Even like generally what we'll do is we will, this is a 1 in 100. Our working concentration or our working counting density or dilution is a 1 in 200 dilution ratio. Getting this down to a 1 in 200 would probably be fine. But if it was still too dense, you could cut that. You can't tell if it's blurry or the stream is blurry. Yeah. So this is, there we go. Yeah, this sucks. Okay. So I don't know how much actual cell counting I'm going to be able to do. I'm probably not going to be able to move around a lot, but as you can see here, there's just a lot of yeast on here. So this is a Cali strain. As we can see, there's not a lot of, they're not all clumped. They're not all clumped, which means that this is pretty easy. This is pretty easy to count. I'm going to move over to our English strain, and what's really funny is I guess I roused it and shook it up enough that it didn't clump up. But generally, whenever we're working with English strains, they're extremely difficult to count. So we have to actually treat them with a chemical. And this is this is English. Unfortunately, it's a bad example of what it would typically look like, but we can see a little bit in the. Just let me move. So if we're looking at this and we take a look at the third section, we can see, and then so in the middle of the screen, there's the three areas. On the third one in the middle, there's like a clump of yeast there. That's typically what we'll see with English strains. And generally, they'll end up clumping on top of each other, and it makes them extremely difficult to count. So right now, this is an untreated sample. So with that, all we can do is do a cell count. We're like, this is the density of the yeast, but we don't know whether or not that yeast is alive or dead. So what we do is we treat them with some kind of dye, and then the live cells will either prevent the dye from entering or they'll excrete it, and then the dead cells, the dye gets into them, and then they just they just stay that color, and that's how we can tell whether they're alive or dead. So this is just how we navigate around. Unfortunately, the zoom on my scope camera is really high, so I can't zoom out any further. But I will zoom in a little bit closer. I think we'll be able to do that. And maybe we'll be able to get a better picture of the yeast. Okay, so there we go. This should be a little bit easier to see for everybody, and I'll just bring that in focus. All right, so this is yeast. This is an English ale strain underneath a microscope. So that's typically what we'll be looking at. At a normal 40x or 400x, it would be zoomed out a bit further, so you could actually see all edges of this counting area. And then I'll just move over to the Cali, and we'll see how much of a night. So this is what it would look like counting Cali. That's really funny. So there was some extra methylene blue on my counting chamber. That's why these cells are picked up. These yeast haven't actually been treated with anything. So always make sure that you clean your counting chamber really well, so you don't get stain pickup on your yeast. So this is what your yeast will look like underneath underneath the scope. All right, so I'm going to zoom back out again, and we will just go back into focus on the presentation. So dilutions. As I mentioned, those were already pre-diluted. Whenever you're performing counts, on a yeast slitter, you need to dilute the sample in order to get to a density that's reasonable to count. And then you can use that number to estimate the actual cell count. Internally, what we're looking for at the lab, we generally want our cell counts to be whatever we're counting, whatever our concentration at. We want to see over 70, like over 75, and generally under 400. Whenever you get over 400, there's going to be so much on there that you're going to start making counting errors. Under 75, there's generally not enough on there, and what ends up happening there is you aren't getting a good representation of the population. So it's better to step down your dilutions in order to get a more accurate representation of the population that you're counting. Yeah, you can't just pull up a sample and load it onto your hemocytometer. It's way too dense. You won't even see anything. It'll just be like a carpet of yeast. similar to that 1 in 100 of that really concentrated kali I was showing you. It'll just be worse than that, so you can't even count it. Something to remember, especially if you're working with if you're working at things such as you're working with these 10 mil pipettes, all your equipment does have an inherent error to it. So always keep that in mind. And then there are also techniques that you need to do to make sure that you aren't also introducing basically technician error into it as well. So always be mindful of that and work around that whenever you're setting it up. So when we're doing dilutions, to perform a dilution, you're going to take a small sample of your dense yeast solution and then dilute it in water in a calculable manner. So for the stuff whenever we're working with our harvests and our off airs, as we call them, so yeast in suspension in tank, we'll do 1 in 200 for a thick slurry, so that's a harvest for us, but that can be a commercial pitch, the bottom of a tank, a yeast cake for a home brewer, or a decanted starter, and then a thin slurry will be a 1 in 20 dilution. And a thin slurry would be a starter. So let's say you have a starter growing and you haven't let it crash yet. That would be your thin slurry. You could pull off a couple mils of that and then do your dilutions off there. Fermenting beer, or let's say you're doing propagation in-house, you're propagating yeast. So your dilution ratio is the volume of your slurry divided by the volume of your slurry plus the volume of your water. This can also be weight. So let's say you're doing 1 gram of slurry divided by 1 gram of slurry plus 9 grams of water. Working with weight is something you can do that is, it's easier to work with, especially if you're pitching out of things like brinks and stuff like that, where you actually can't see the volume. So that's always something to work with. Also, weight works really well if you're working with yeasts that are really foamy. So you could have something that looks like it's 12 liters of slurry, but it's only 2 liters of slurry, but there's so much foam and stuff taking up all that space that it looks like you have more. So depending on what you're doing and what you're working with, using weight might be a better alternative for you. Whenever I've done stuff at home where I don't have access to all my fancy lab equipment, I've used I like to call it my Coke scale for my salts and stuff like that. And I've just done my samples using that and then doing weights on there. So it works. There is some error inherent to that as well. And whenever you're working with things like that, sometimes the best thing to do is do your dilutions at larger volumes. So let's say you don't have the ability to work at 1 mil. What you could do, or 1 gram, what you could do is you could do 90 grams of water or 90 mils of water, and then 10 grams of slurry or 10 mils of slurry. By scaling up, you're reducing your error because based on the equipment you're using. So that is always something to keep in mind. All right, so here's just an example of what a dilution would look like. We have a 1 in 1 dilution, which is our slurry. We take 100 microliters of that and then add that to 9.9 milliliters of water. So that's 0.1 milliliters divided by 0.1 milliliters plus 9.9 milliliters. That gives us a dilution ratio of 1 in 100. And then we take 10 microliters of that sample and then mix that with 10 microliters of our erythrocin B dye to give us a 1 in 200. So that's 1 divided by 1 plus 1, which gives us 1 in 2, and then we carry it over. So 1 over 1 in 100 times 2, or times 1. And anyway, it's fraction math. So a live dilution demo. I've done my dilutions already. I'm going to show a dilution proper technique. First off, so I need to switch my camera again. So this will be fun. So I have a DSLR running to show some bench work. Should be this one. All right. Okay, so let's focus on this camera. Now, the quality of this stream isn't very great. This is set up, I'm running it through a USB, like a USB port. So the quality isn't great. You'll be able to see the work happening. It's pretty artifacted. I'm sorry about that, but I'm gonna mostly show the technique, and you should be able to get that from this. So whenever we are doing lab work, make sure you're wearing gloves. Now, the work I'm doing, I'm not using aseptic techniques, so the samples are getting destroyed. If you're gonna be working with these samples, for, let's say you're going to be doing quality work after, what you can do is set up an aseptic environment. So you spray it down with isopropanol, or you can use the Escarpment Labs branded alcohol. It doesn't smell good, but it works. And you could spray that down, and then what you'll do is you can use an alcohol burner or a Bunsen burner, and then you'll light that, and then you have this aseptic environment where it protects your samples. I'm not working with a protected sample. So basically this sample, I can't use it for quality or anything like that. I could hold on to it, but I'd note that it's not aseptic, and it can only be used for cell counts. I can't really use it for anything else. So what I'm going to do here is I'm going to show you how to work with your first sample, and then I've prepared some 1 in 100 samples already. So I'm going to be doing a 1 in 100 or 1 in 10 dilution because I am working with equipment that doesn't allow me to do smaller dilutions. So I will write on. It's always good to label your stuff 1 in 10. So I've got my dilution tube that I'm going to be diluting into. You want to rouse your sample really well as you can. And then I'm going to take a clean pipette tip and pull that sample. Now, something to be aware of here is, especially with thick slurries, is you're going to end up picking up a bunch of yeast on the outside of your pipette. And you want to make sure that you get that yeast off. Otherwise, it's going to introduce a higher than expected cell count because you're bringing in more than the sample you think you are. So what's best to do with that is take a Kim wipe and I'm getting a bunch of stuff falling off. So another thing to be aware of is these pipette tips are low quality. So I'm definitely not going to get a 1 in 100 sample in there, but we can correct for that. So anyway, wipe your pipette tip off. This gets all the yeast off of it. And then you will take your sample, insert it into the water, and then you're going to pulse it back and forth to rinse off the inside of your pipette to get the rest of the slurry out. And then you can dispose of that. And then you can shake it. If you're doing a lot of cell counts, I strongly suggest getting a tool such as this, which is a vortexer. We got this one from Fisher Scientific. We ran it through basically the gauntlet at Escarpment Labs, and this one is pretty much broken. We can use it for some things, but it doesn't work that great. And then you'll just vortex it to homogenize. If you aren't getting a good vortex, you can put it sideways and then vortex it sideways as well. But if you don't have that, you can just shake it. Make sure you shake it really well. Now this is a 1 in 100 sample. This is still pretty turbid, so I still wouldn't be able to work with that. So eventually you'd get a dilution down to a 1 in 100. This is still pretty thick, but I'm not going to worry about it because we're just going to do the dye after. So this is a 1 in 100. Now what I want is I have these prepared samples of erythrocin B. So this is 1 milliliter of erythrocin B. Another thing with working at lower volumes with, say, a 100 microliter pipette or a 10 microliter pipette is you end up using less stain. So you'll end up, let's say it costs you $35 to buy your stain and you only get 100 mils. If you're doing 1 mil tests, that's only 100 samples. If you do 100 microliters, that's 1,000 samples. So you can make it last longer and you can reduce your costs. So again, I'm going to take my 1 in 100 sample. I'm going to shake it up, make sure everything's nice and in solution. And then I'm going to pull a 1 mil sample, and then add that to my 1 mil of erythrocin B. So again, I'm going to collect my 1 mil. Take my dye. Add that. You can pulse it back and forth, especially when you're working with smaller concentrations like a 10 microliter pipette tip. You'll want to do that. And then you will vortex it or shake it to fully homogenize that sample. For loading your pipe, this is where working with these 1 mil tips is a bit of a pain. It's going to be really hard to load onto the chamber accurately. These chambers are designed to take 10 microliters. And if you load more than that, you'll end up flooding the chamber. And if you already have it, and what you can do is you can load 2 samples on this. So if you want to, if let's say you're counting 4 harvests, you can shorten your time by loading 1 sample onto each. But if you end up overloading it, the 1 sample can bleed into the other, and then you've contaminated both of your samples. So you'll need to redo it. Now let's hope I don't screw this up and don't end up having to do that. So pull a little bit of your sample, your fully homogenized sample, and then using capillary action, you're going to load that onto the chamber. So what's going to happen is I'm going to put a little bit of liquid on there, and then there's a small gap in between the counting chamber and the glass. And what happens is I load liquid on there, and then it sucks the liquid in. You don't have to force the liquid into the counting chamber. The capillary action on its own will do the loading for you. All you have to do is make sure that you get the liquid onto the glass right beside it, or right beside the cover slip, and the counting chamber or the capillary action will pull it in for you. So I don't know if you'll be able to see that really well, but this is what a loading counting chamber with dye looks like. Let's put it underneath the scope, and I'll just get it in focus here, or I'll. So I'm just going to find. We're not going to look at everything. Okay. Now again, I need to go and I need to swap my camera so we can look at the. Come on. So we can actually look at it underneath the microscope, and now we're going to be able to see live and dead yeast. All right. Okay, now I'm hoping there's some dead. Oh, there we go. There is one dead yeast in there. Sweet, perfect. Right. So this looks pretty good. So this is a 1 in 200 dilution. You could count this. You could totally count this and it would be fine. I'm just going to hunt around. I'm going to zoom in a bit more and try to hunt around for a better chamber. Or a better part of the chamber. Oh, here we go. This is a good one. So let's just zoom out. Oh, there you go. There's a really up close view of some dead yeast. All right. So as we can see here, even with the quality of the stream, you can still see the dead yeast. So what's going on here is the dead yeast are unable to excrete the dye and or metabolize it, or however it's working. It varies depending on the stain, I think. But what we can see here is we can tell which yeast are alive and which yeast are dead. So what we can use that information with is we do a cell count. Generally, if you shouldn't have to use 2 counters to count live and dead yeast. There should be enough dead yeast, or there should be not so many dead yeast that if anything, you're just writing each chamber or each section that you counted on a piece of paper, or you can count it in your head. I think usually we generally won't see over like 20 or 30 dead in like the worst case. But if you're getting higher than that, like let's say you're working with homebrew or something like that, or you're working with a really old pitch of yeast and you want to verify that, then you'll want to use 2 counters or count all the live ones, then count all the dead ones. for a really good, really viable yeast, you should expect to see something like 95% viability or higher. Depending on how you treat your yeast, at like a homebrew level, I harvested some Cali that I had used to make a pale ale that I was going to make a barley wine with. I pulled that off and I treated it like shit, and that was at 80% viability. So that was still pretty good. Like considering this was yeast that had gone through a fermentation, an anaerobic fermentation, and was not really treated well. And the only thing I did was, once that beer was done fermenting, I pulled it off the yeast almost right away. That's all I did. But if you're moving pretty fast on your yeast, you should be able to be within 70 to 85% viability at a homebrew level. On a pro level, you can probably get it higher because you have a lot more control and stuff like that. And yeah, so that's kind of what's going on here. This is what things will look like. If you're using methylene blue, whenever you do your staining, you need to let it cure for about 2 minutes, I think. But you can't just like put the dye in and then take a sample and load it onto your counting chamber right away. What you need to do is you need to let it sit there so that the dye can do its work. The nice thing about Trypan Blue, which we don't use anymore because it's really bad for the environment and people, and erythrocin B, is that the dyes work almost instantly. So by the time you're done homogenizing everything, you can load it onto your chamber right away. So that's kind of the nice thing about working with these. And again, erythrocin B it's just a food stain. So it's also a lot safer to work with, and it's safer to dispose of. So that is the dilution demo. Let's get back to, I didn't kick him, did I? Oh no, I didn't. Okay, let's. Oh no, I think I booted myself. How do I bring him back? Okay, well, we'll see if we need to do that. But let's just keep going. Oh, I did boot him. Okay, let's bring him back. Before we get, I'll just accept and join, do all that, blah, blah, blah. Okay. Okay. I just want to make sure that whenever the video feed for the camera comes back, it doesn't. All right. Anyway, I'm not going to waste more time on this. So, as I showed, refresh and try again. Okay. As I showed in that demo, the final dilution step is 1-to-1 with erythrocin B. This is loaded onto the Hemocytometer, and then 10 microliters of that sample is loaded onto a cover slip. Load your 10, and then placed onto the counting chamber. Angling the pipette can assist with proper loading, and you can use a pipette, a disposable pipette, or an eyedropper. Pasteur pipettes are those like bulb pipettes that you can use, and they're very, very cheap. You can use those for loading onto your chamber as well. Once loaded, we want to find the proper counting region and begin counting. So this is what the region looks like. We're going to find that center chamber, and then we're going to, you can do this zoomed out and then zoom in. Find that counting chamber, zoom into it, and then count all 5 regions. And then whenever we're close in on our counting area, what we're going to do is we count all of the yeast. But the thing is, we don't count the yeast everywhere. So basically, we have a rule that whenever you're doing your counts is to keep things consistent between lab technicians, you don't count the right, any yeast that fall more than half or 3/4 into the right or bottom border, and those borders are signified by the 3 lines. So each region will have this 3-line border, and then anything that's on the right or the bottom, we don't count, and then anything that's 3/4 or more on the top or the right, we will count, and then anything else in between, we count as well. So that's how we begin counting. Count all of those, and then, and to properly count, you need to count, as I mentioned. So in this stream, that is anything with a check mark on it. Those are ones that are okay to count. And then anything with an X are ones that you exclude from that count. Do that for all 5 of the grids in the counting matrix, then we can do some math. So I showed this already in the video stream, but here again is just a picture to show you of what dead yeast look like, and then what protein, and also there are other things will come up on there. So you will get protein and trub that show up. Yeast look like little circles. If it looks like, sometimes starch will look like little squares, and trub and stuff like that looks like just a bunch of stuff. It's just a bunch of protein all folded in on itself. So that's generally what it'll look like. That will come up because depending on, like, especially if you're bottom cropping, maybe you have like trub and stuff that's also mixed in there, depending on just how things flocked out. And then, yeah, you'll use your counters to keep track. And then dead cells will appear the color of the dye that's being used. So in the case of erythrocin B, they'll be pink or red. And a good rule for this is, like, if it shows any color of the dye, this is more of a problem when working with methylene blue. Sometimes the yeast won't go fully blue. Like, they don't go nice and, like, the nice thing that we liked with Trypan B is, or erythrocin, or yeah, with Trypan blue is that, like, the yeast would be like 100% blue all the time. And we've also seen this with erythrocin B. The yeast will be fully red. The counting chamber demo, I technically did that already, so we're going to pass through that. But again, just remember, that's what it looks like, identifying live and dead yeast. How do you clean your pipette? We can, if you want to ask that question in the ask question section, we can get into that after. So this brings us to topic 4, which is our math and our calculations. So when you're finished a count, it's time to do some math. The equation is dependent on the dilution rate performed. First thing we want to do is we want to calculate our viability. All that is we just take the total number, the live cells we counted, and divide that by the live cells plus the dead cells. Again, it's a ratio. So similar to a dilution, it's a ratio. So all we're going to do there is we're going to take our live cells and then divide it by the sum. So if I counted, let's say I counted 75 live cells and 25 dead cells, I do 75 divided by 25 plus 75, which gives me 0.75 or 75%. That is my viability. Now, if we want to calculate our density, which is cells per milliliter of slurry, we do 5. This is a simplified formula. If you want a full breakdown of the formula, I think it's covered on the Braukaiser blog, and also the yeast book by Chris White also covers this formula. And then it's all over the place. You can easily find it. This is a simplified formula, so you're literally just plugging in blanks. You're doing 5 multiplied by the live cells that you counted, multiplied by 10,000, which is 0.1 microliters in the counting area adjusted to milliliters, and then your dilution factor. We say from our previous example, in an example, let's say we had 21 alive and then 20 dead, we have a 51.2% viability, and then our density, assuming a 1 in 200, would be 5 times 21 times 10,000 times 200, which gives us 2.1 times 10 to the 8 cells per mil, or that's 26. No, that's 200 or 210 million cells per mil, I think. Components are hard. Viability and density can be used to estimate how much of your yeast need to use on your next pitch or for your next brew. Repitching is where the cost savings come in and can offset the cost of investing in cell counting equipment. If a sample has poor viability, it's best to find fresher yeast from within your cellar or to contact your supplier to get new stuff. Something to do here is you can try to do your counts far enough in advance that you're not freaking out and rushing around trying to find better yeast. And also it gives you time to actually have your supplier get you the yeast and ship to you. So, and another thing to do here is while you're doing your counts, every time you you do a harvest and do that stuff, track it somewhere so you can actually monitor your trends. And then you can see either because of maybe nutrient issues or fermentation problems, you're seeing a massive drop in your viability. And then you can start predicting based on what kind of beer you're making when you need to start ordering fresh yeast. Then you can build that into your planning and make sure that by the time you get your fresh yeast, by the time you need fresh yeast, it's already been ordered. So the math, no one wants to do the math, and the math sucks, and we get that. So if you need to calculate pitching rates, you can go to our website. We built out a really simple pitching rate calculator. You just go to escarpmentlabs.com/cell-counter. We have a pro and homebrew version. The only difference is the units that they work with and the number of cells that it displays to you. It's the same math underneath the hood, except it's just a scale has been adjusted. And then we do things for, on the homebrew side, we do things in liters and gallons because that's what people work in. But you can fill in everything here. And then if you bring down that advanced column, you can put in what your pitching volume is, what your desired pitching rate is, and all that kind of stuff. So you can play around with a lot of these things on our cell counter or on our pitching rate calculator / cell counting tool. So effectively counting yeast for English-like strains. So I have here some English one, but it isn't clumping as nicely as that gross stuff does. But English strains generally don't flock very well, or you can't deflock them really well. They'll reclump again. So some of the problems with that is it makes it really difficult to pull a homogeneous sample, which may result in cell counts that are too high or too low. These strains can be treated with an organic acid, such as phosphoric acid is kind of the industry standard for that, to discourage yeast from clumping, and this allows for an accurate sample to be collected. If the samples you're working with are too foamy, you can also treat them with this compound called 1-octanol to defoam it, and you only need a small amount. So we use our Foggy is extremely foamy, and it's really hard to get accurate counts on. So we've integrated using 1-octanol, which it's like I don't even remember how it works, but it basically prevents the foam from forming, and then it makes it easier to pull more accurate samples. Because sometimes we pull samples, and then half of our pipette tips are foam, which means we're not getting what we think we're getting. So 1-octanol is another thing you can integrate into your process to make it simpler. So I'm going to switch back over to the So anyway, I'm just going to show this up here. We can see that we have this English strain, which is pretty clumpy and gross. What I'm going to do is I'm going to use 75% phosphoric acid. I'm going to add half a milliliter, which is way too much. One of the things to be aware of when working with these acids is they are going to increase your. So I'll add 3 ml or 0.3 milliliters of phos. Another thing when doing stuff like this is when working with these, they are dangerous. So wearing gloves and PPE is appropriate because you don't want to get them on you because you can get chemical burns. So again, just dose that in there. I'm going to clean this off my desk, and then I need to remember to wipe it down after. And then you can just vortex it or shake it. But again, something to remember is the yeast. This is killing the yeast, so you don't want to use a lot of it. I think for 500 microliters of Foggy or English or whatever, we add 3 microliters of phos, and if you work fast enough, that is good enough to get you yeast that you aren't completely hosing it so that you don't get a good enough sample, or that you get like artificially elevated death rates. And now we can see the sample isn't clumping, isn't flocking anymore. So we can actually pull a more homogeneous sample and get a better cell count from that. When doing stuff like this, it's always best to, especially if you're going to be using it, if you're either going to be doing future cell counts or you're going to be working with it in the if you're going to be using it for QC, always pull a sample off, because this is a destructive this is a destructive procedure. So pull a sample off and then work with that sample you'll be destroying, so you don't compromise the integrity of the original sample. Especially true if you're going to be doing QC on it. Yeah, so I've kind of talked about this already. Agitate the slurry to fully declump it, quickly move the slurry into your dilution tubes, and then perform your cell count and viability checks. Generally what I'll do whenever I'm counting foggy or English-like strains is I'll pull my sample, faucet, get it homogenized, and then quickly do my dilutions and load it onto my chamber. I don't like, for other things, you can do your dilutions, you can prep all your dilutions. So let's say I have 6 things to count. I would prep all of those dilutions first and then do my actual staining and counting, because then it just makes my process easier. But with foggy and English strains, you don't want to leave them in contact with that phosphoric acid for too long, because what it does is it ends up killing your yeast and giving you a bad cell count. So with working with foggy and stuff like that, the thing is to move fast on the sample, which will slow you down overall, because you're just swapping between jobs a lot more frequently. And again, you can, if your viabilities are really high, you can double check the viability by staining an untreated sample and then checking for dead yeast. So that's more of a spot check. So let's say you got like 75% viability. You're like, well, that can't be right. This is only my first generation. Pull another sample, treat it as you would a normal yeast, and then just do a stain on it and look at that. And then you just do like a quick visual check and be like, yeah, this is like not really 70%. It's fine. Then you can like ballpark it, or you could do a cell count. But always remember that whenever you don't treat them, those yeast will clump on top of each other, and you can end up with these like clumps that you can't even really count. So it's always something to be aware of. That was my chunky yeast demo of like treated and untreated yeast. So this will bring us to the questions and comments section. So I'm gonna kick out our camera from this, and I'll bring Alex back. Boot. And then I will bring back Alex. All right. Oh, and surely all of you have learned how to properly take gloves off, but let's cover it anyway. So whenever you're taking your gloves off, you've always got to remember your gloves are contaminated, or treat them like they're contaminated. So you put your finger underneath, pull the glove up, and then put it in your other hand.
Am I back?
Hey, Alex. Yeah, you're back. Put it in your other hand, and then you go underneath, and then again, treat it like that. And now you have a clean surface on the, you're working with a clean surface, and all the dirty stuff is on the inside. This is really important especially if you're working with chemicals such as phos and stuff like that, to keep all the stuff that could hurt you or someone else in, keep all that stuff inside the glove. So just proper glove removal as well. All right. Oh, Alex was back. Now he's gone. Oh, he's reconnecting. I can close this. I can turn this camera off. Hey, Alex. All right.
Chris, can you hear me or no?
I can hear you, yes.
I cannot hear you, so I don't know what's wrong with mine. And I have no idea what to do.
All right. Can you hear me now?
All right. Well, Chris can hear me, but I can't hear Chris. So I'll just read his lips. So thanks, Chris, for the informative talk. It was great. It was a full packed hour. So we do have time for questions. We've answered some questions in the discussion forum itself, but there is an ask a questions area. So if you do have questions, head on over to that, upvote the ones you want answered the most or that are the most pressing to you. But before we get to that, I just want to let everyone know that we are having 2 more sessions today. One is at 12 with myself. I will be talking about, if you don't have a lab, some tests you can do with equipment that you should have around your brewery. And that's at 12. And then later, Diastaticus 101 with Nate and Richard at 2 p.m. So we can start answering some of those questions, assuming everyone else can hear Chris. So the first one is, what do you think about automatic cell counters like Oculize?
So if I can recall correctly, I believe the Oculize uses, it's like a little thing that you attach to your phone, and then you put your sample on a counting chamber, and then you count it. It's really funny because I come from software, but I'm actually really skeptical of anything that requires apps, because it's really easy, especially with like vendors for stuff like this, is generally their apps are really bad. And also you have no guarantee that this piece of equipment you buy is going to work 5 years from now. Like, sure, 5 years is a long time, but like, let's say you're spending a couple $100 on this. You don't want this thing that you're using to, like, let's say they don't update it for the latest version of iOS or something and it breaks it. You don't want this piece of equipment that you bought that maybe by the time it is no longer supported, you are unable to, like, you've highly integrated it into your process and it's really important, and now you have technicians who really don't know how to do counting or are really rusty at counting. So whenever it comes to stuff like that, I am not a fan of sort of like those boutique app-based solutions. There's one that we were looking into, but I don't remember what it is. I don't remember what it is, but there's another one that we were looking into that is a counter that we were going to use, but that's kind of on hold right now. But yeah, that's just the way it is for us. And also, maybe there is artificial intelligence statistics are getting used to count and analyze thousands of samples at once. But a lot of these boutique samples they're still extremely manual. So I don't really see it. And also, there was one we looked into, and it was like it couldn't even work with yeast anyway. We were told it could. We brought it in, it couldn't count them. So that's something to always be aware of. But I know that the Oculize, their big selling point is you can count yeast with your phone. But that's kind of my opinion on it. Hopefully, we will get there eventually. But it really comes down to how many cell counts are you doing. If you get good at it, like once you get enough practice, you can be ripping through cell counts in like 20 minutes. So and like by 20 minutes, I mean like lots of samples in 20 minutes. There is always a learning curve to things. So yeah, but hopefully we'll get to a point where we get tools that are reliable enough and work for that. So that's really not an answer, but that's my opinion on it. All right, Alex, can you hear me?
So I still can't hear you. I'm assuming the question one is done.
Next question.
Yeah? Okay. Cool. So the next one is, do you have any personal recommendations on a microscope and hemocytometer for a home brewer budget? Also a good source of lab supplies and, for example, stains for home brewers as well.
Yeah, so in terms of stains, methylene blue is pretty hard to, at least it seems in Canada, it's pretty hard to get now. I think you can still get it, like as someone mentioned, you can probably get it off Amazon and eBay. Those are probably good places to look for getting those dyes. Erythrocin B, I think you can probably just find a food dye that works with that, and then you can work with making a dilution that works there for you. in terms of a microscope and hemocytometer, this microscope that I was using, I think it was $400, and that's pretty good in terms of equipment that you can get. You can go lower. But they generally start to lose features at that point. I have I think it's a $250 one, but it's a monoscope. I can't hook up my camera, and I can't use the eyepiece and the camera at the same time. And it doesn't have a stage that I can move around. So you can get lower cost ones, but that does come at features that are kind of important. So I would suggest aiming around $300 to $500 is probably what you're going to be looking at. You can go lower, but just know that it comes at a cost of features that are kind of important. And in terms of a hemocytometer, as I mentioned earlier, the one that I have is I don't know if this is Neubauer improved is what I've seen on all the ones we've used. Those are the ones that we use, and they're about $125 to $150. I wouldn't suggest anything less than $100. You go under that, and you're getting low quality counters, generally like low quality cheap lab counters that are imported from China. They do work, but they're not great. I would strongly suggest against that. Even though it does look affordable, you get what you pay for, and it's not worth it. All right, so the next question, Alex.
Yeah, okay.
Yeah.
Yeah, I have no idea what you just said, but I'm sure it was great. I did see you pick up a hemocytometer, and I have no idea what you said, but I know Richard did actually provide links to microscopes and hemocytometers that we recommend in the chat. So assuming that that's kind of what Chris was saying, there might be more information there. We'll answer a couple more questions. Yeah, two?
Yeah,
Yeah.
How much, do we,
Three,
when
maybe?
are we? You can't hear me.
The next one is, in order to determine cell density from fermenting beer, is it necessary to fully degas the sample?
Yes. The reason why you need to degas the sample is you need to make sure that you're pulling the proper sample to begin with. You can always verify what the sample you're collecting is at the right. It's so annoying. It's so bad. You need to verify that the sample is at the right volume you think you're collecting. You can always check that. But depending on the type of beer, you might not be able to tell whether or not it's gas or liquid. This comes back to that 1-octanol chemical you can use. You can add 1 microliter to a sample, and it will basically instantly degas your sample. So you can use that, or just shaking or vortexing will also really help degas the sample. But degas your sample, make sure all the foam is gone. Spin it, burp it, spin it, burp it, do that a few times, and that should get all the CO2 out of solution, and then you can pull your sample. So yes, always make sure that you're working with a degas sample as much as you can. Otherwise, you're introducing error. Next question.
Yeah, okay. Oh, this is so weird. I'm sorry. Yeah, I have no idea. The next one is there a phase of the cell cycle that affects yeast's ability to break down or metabolize dye?
That's a good question for Richard. Richard can answer that in the chat. Unfortunately, I do not fully know, so I'm not sure about that question. But hopefully Richard can answer it in the chat, and then we can talk about that after. So we will go to the next question.
So I saw the word Richard and not sure.
Yes.
Yeah.
Yes.
So my answer is if it's healthier yeast, typically they have more capability to break down dye, depending on the type of dye you're using. So certain dyes act if they're alive. Some just fully penetrate only dead yeast. I think it's methylene blue enters all cells, and then if it's a healthy cell, it can utilize up to methylene blue. So it depends on yeast health sometimes, but I also agree that Richard is totally the person to to properly answer that question. Yeah. We'll do one more because I think it's important to really drive home, and then I think we might call it quits if you're good with that. Does the microscope with a 400 times objective, is that good enough to see bacteria or not?
So you can see it. You can play around with your phase a lot. Also, another thing of getting quality microscopes is they give you this thing. It's you can play around with the phase, which plays with I think it's the angle that the light goes in, and you can see. So at 400x, you can see bacteria underneath the microscope. You really can't make too much of a judgment on what it is, though. You can be like, oh, that looks like a cocci, or it looks like a bacilli, but you can't do a count on your bacteria, but you can see it. And one good thing that doing, let's say you want to verify your bacteria underneath the scope. This is good for harvested bacteria, for harvested sour cultures, such as a kettle sour culture, and validating that it doesn't have Saccharomyces in it. So what you could do with that is you pull your sample off. Let's say you're going to go use it for another kettle sour in 2 days. You shake it up, pull a sample, and then put that underneath your microscope. At 400x, you'll probably, if it's dense enough, you will see bacteria under there, and they'll be jiggling and stuff like that. But the big thing you're using that for is to make sure there's no Saccharomyces in there, and those things are huge. So you'll see them, and you'll be like, okay, my sour culture or my kettle sour culture is contaminated with sac. I can't use this. So it can be good for a spot check. One thing to be aware of, though, is if you're going to be doing this, make sure that you are doing all of this aseptically and you're using clean equipment and sterile equipment. Because if you aren't using sterile equipment, you run the risk of introducing a contaminant from your equipment into your sample, and now you're getting a false positive. So always make sure that if you're going to do this kind of stuff, that you are using preferably disposable lab equipment, such as disposable pipettes and stuff like that, so that you know that what you're working with is sterile to begin with. All right, and we're done.
Good. Okay, that was a nice long answer. I'm sure it was really great. Awesome. So we just wanted to thank everyone for attending today's webinar. I have no power, so we cannot end this, but thank you, Chris, for putting this on.
No problem. Thank you, everyone, for attending.
All right. Bye-bye.